Guys, now the topic is DNA fingerprinting. How DNA fingerprinting works? First of all, it was proposed by or given by Alec Jeffrey. DNA fingerprinting was proposed by Alec Jeffrey. He concluded that 99.9% of DNA sequence, or we can say base pairs, is same in all individuals. Base pair sequences. Okay, means the differentiation between 0.1% base pair of individual different. Means if we are going to talk about the individual base pairs, means मेरे अंदर आपके अंदर हम सब के अंदर 99.9% बेस पेयर सिमिलर है, तो डिफरेंस क्या हो गया? ओनली 0.1% बेस पेयर जो है वो हमारा अलग हो गया. डीएनए separated on density gradient method, or we can say centrifugation method, as we have talked about that. We have two ways to identify DNA fingerprinting. The first one, we analyze only coding or a different sequence, or second one, we analyze a whole sequence. So we just going to go through with study of a whole sequence. It provides two segments, just to be like that. It's a bulky group and this one. It's a bulky major group is 99.9%, means common in all individuals. And this small peak is 0.1%, means different in different individuals. And this DNA is called satellite DNA. And this DNA is known bulk DNA. In a satellite DNA, it has a repeated sequences. In a satellite DNA, I mention it here like that. That's a micro satellite DNA, means this DNA sequence are larger and mini satellite DNA means these sequences are smaller. In mini satellite DNA, it is also work as a VNTR. Remember the term VNTR means variable number of tandem repeats. Number of tandem repeats. VNTR actual used as radio level probe. Guys, here probe means a strand of DNA or a part of DNA. It is useful to identify DNA fingerprinting technique. We categorize silent DNA also or a satellite DNA in a microsatellite or a mini satellite. There are some classification by which we categorize that, and these classifications are, I just noted here. It is first purine, pyrimidine basis. Second is length of sequence means mini and microsatellite, and repeated sequence, or we can say copy number, how many times they can copy it. Next point is the method how can we identify with the help of the method, and was the procedure to getting DNA fingerprinting. First of all, isolation of DNA. Isolation DNA means we just analyze which kind of DNA or on a which cell we want to be DNA. So just extract the DNA from that cell. Second step, break down, break the DNA, and as we talked about, we break it with the help of endonuclease enzyme by endonuclease enzyme. Third is separation by gel electrophoresis. In gel electrophoresis, it's a instrument. We know DNA carrying a negative charge, so we apply negative charge here, and on other hand apply positive charge. It is filled with a matrix or a medium. The whole chamber filled with a matrix or a medium, and this matrix or a medium is called agrogel, or we also says that agar हाँ agar medium, and this agar medium provide to separate out the DNA sequence. We just make some holes and put the DNA here. We filled with DNA with endonuclease enzyme. Means endonuclease enzyme work as it's a DNA sequence, double helix structure of DNA. It cut the DNA from various parts by its identification sites. After that, we just apply electric current or electric field here. DNA carrying a negative charge, so it go away from that side and attract it toward its opposite side. We just assume a DNA sequence with a large DNA or a base pair sequence and a DNA with a less base pair sequence. Means it's a lighter DNA or it's a heavier DNA. So lighter DNA move faster as compared to heavier DNA. So lighter DNA it should be there and a heavier DNA it should be there. By this we apply here ethidium bromide exposed in ultraviolet radiation or sun rays. It appear as means DNA sequence appear as orange color bands. We extracted it from agarose gel by wash with chilled ethanol. So we get extracted DNA sequence easily here. Now our fourth step is blotting. Blotting means nitrocellulose membrane. We just wash it with a nitrocellulose membrane easily. Fifth one, hybridization with radio radio labeled means having radioactive nature. VNTR variable number of tandem repeats. And the next is auto radiography to identify VNTR. And these VNTR just to be alike as a band. We use this technique maximum in forensic science. Means we assuming there is a suspect A and a suspect B. There are two suspects and we got a cell sample from the victim site, a dead body having a cell sample, and we have two suspect A and B. We don't know really that the who were present on the crime. See, so what we have to do? We just take a cell sample of A, cell sample of B, and we have cell sample. We mark it as C from the crime scene. We just analyze. We just analyze the sample of crime session is maximum matched or matched with suspect A. So we analyze that on the crime session on that time, suspect A was present and suspect B is relieved. So it is used in such kind of things. The second one is, we take another example, that's a child. They are claiming by two parents, that's a father F1 and a father F2. He is saying that the child is related to F1, or he is saying child related to F2, means they have two fathers, F1 and F2. But scientifically, we just used this technique, or it's called paternal test, means we take genome or DNA sequence of F1, DNA sequence of F2, and DNA sequence of C, and by which whom it maximum match, then the father is that of that child. Guys, now the next concept is operon concept, or we can say lac operon. Lac means it is related to lactose. Operon operate. Now lactose is able to operate. How can we do? Means we are just going to make an RNA sequence, or we can say it's an lac RNA. How can we do it? It was firstly proposed by Jacob and Monod. So we can say it as Jacob and Monod concept. It has two types. Operons are of two types. That is, first is inducible operon. Second is repressible operon. Before we are going through with inducer and repressor, we just know about some other things, like I'm just going to make a diagram here. Here there is a gene I. I'm mentioning it here, gene I. It form repressor. Repressor. It form repressor. Next is three functional or structural genes here. Structural. Z, Y, and A. Here Z act as a beta glycosidase. Y work as a permease, or A work as a transacetylase. We mention it as A's word, means these are the enzymes here. So what we have to do in this? It's the inducer system. First of all, this one started here. Stable. operon, or we can say it's a switch on. In this process, I make an mRNA strand that is called repressor. As we mentioned here, repressor. I make an mRNA strand or a repressor. It form a protein molecule. This is repressor protein. It's a repressor protein. Here another protein, that is inducer protein. Inducer protein. It's an inducer protein. Inducer protein bind with repressor protein and make it inactivated repressor. We can say here inactive repressor. Now these genes, these structural genes make another, wait, there is a O, operator, another mRNA, mRNA, and this mRNA used by lactose. So we can say it as lac mRNA. Here lac mRNA Z work as a glycosidase, glycosidase. Here glycosidase work to break down lactose into glucose plus galactose. Y enzyme permease permit glucose and galactose inside the bacterial cell, and we used here the bacteria E. coli. It permeates to the entry of glucose and galactose in E. coli cell. Third is transacetylase, which work to make, or transacetyl means formation of protein here. Means now the protein formed from source of lactose. Now in second step, what we have to do? I repeat it again. Here PIPOZYAI is a structural gene which form a repressor mRNA molecule. It form a repressor protein which bind to the operator system, and when it bind to operator system, it just block, or we can say here it just stop the functioning of Z, Y or A, means beta glycosidase, permease and transacetylase. That's why there is no formation of protein. This concept was given by Jacob and Monod.